Breakthrough. Study shows low-carb diet may prevent, reverse age-related effects within the brain. I'm Steve Belair. Welcome to Breakthroughs. A study using neuroimaging led by Stony Brook University professor and lead author Lilianne Mujica-Parodi, PhD, and published in PNAS reveals that neurobiological changes associated with aging can be seen at a much younger age than would be expected, in the late 40s. However, the study also suggests that this process may be prevented or reversed based on dietary changes that involve minimizing the consumption of simple carbohydrates. To better understand how a diet influences brain aging, the research team focuses on the pre-symptomatic period during which prevention may be most effective. In the article titled "Diet Modulates Brain Network Stability, a Biomarker for Brain Aging in Young Adults," they showed using large-scale, life-spanning neuroimaging datasets that functional communication between brain regions destabilizes with age, typically in the late 40s, and the destabilization correlates with poorer cognition and accelerates with insulin resistance. Targeted experiments then showed this biomarker for brain aging to be reliably modulated with consumption of different fuel sources. Glucose decreases and ketone increases the stability of brain networks. This effect was replicated across both changes in total diets as well as after drinking a fuel-specific calorie-match supplement. What we found with these experiments involved both bad and good news, said Mujica-Parodi. The bad news is that we see the first signs of brain aging much earlier than we previously thought. However, the good news is that we may be able to prevent or reverse these effects with diet, mitigating the impact of encroaching hypometabolism by exchanging glucose for ketones as fuel for neurons. What the researchers discovered using neuroimaging of the brain is that quite early on there is a breakdown of the communications between brain regions, the network stability. We think that as people get older, the brain starts to lose the ability to metabolize glucose efficiently, causing neurons to slowly starve and brain networks to destabilize. Thus, we tested whether giving the brain a more efficient fuel source in the form of ketones, either by following a low-carb diet or drinking ketone supplements, could provide the brain with greater energy. Even in young individuals, this added energy further stabilized brain networks. To conduct their experiment, brain network stability was established as a biomarker for aging by using two large-scale brain neuroimaging fMRI datasets totaling nearly 1,000 individuals aged 18 to 88. Destabilization of the brain networks was associated with impaired cognition and was accelerated with type 2 diabetes, an illness that blocks neurons' ability to effectively metabolize glucose. To identify the mechanisms as being specific to energy availability, the researchers then held age constant and scanned an additional 42 adults under the age of 50 years old with fMRI. This allowed them to observe directly the impact of glucose and ketones on each individual's brain. The brain's response to the diet was tested in two ways. The first was holistic, comparing brain network stability after participants had spent 1 week on a standard, unrestricted versus low-carb, for example, meat, fish with salad, but no sugar, grains, rice, or starchy vegetables diet. In a standard diet, the primary fuel metabolized is glucose, whereas in a low-carb diet, the primary fuel metabolized is ketones. However, there might have been other differences between the diets driving the observed effects. Therefore, to isolate glucose versus ketones as the crucial difference between the diets, an independent set of participants was scanned before and after drinking small doses of glucose on one day and ketones on the other, where the two fuels were individually weighed, dosed, and calorically matched. The results replicated, showing that the differences between the diets could be attributed to the type of fuel they provide to the brain. Additional findings from the study included the following. Effects of brain aging emerge at age 47, with most rapid degeneration occurring at age 60. Even in younger adults under age 50, dietary ketosis, whether achieved after 1 week of dietary change or 30 minutes after drinking ketones, increased overall brain activity, and stabilized functional networks. This is thought to be due to the fact that the ketones provide greater energy to the cells than glucose, even when the fuels are calorically matched. This benefit has previously been shown for the heart, but the current set of experiments provides the first evidence for equivalent effects in the brain. This effect matters because brain aging, and especially dementia, is associated with hypometabolism, in which neurons gradually lose the ability to effectively use glucose as fuel. Therefore, if we can increase the amount of energy available to the brain by using a different fuel, the hope is that we can restore the brain to a more youthful functioning. In collaboration with Dr. Eva Rete of Massachusetts General Hospital, we're currently addressing this question by now extending our studies to older populations. Gotta get me some ketones. I'll leave a link to the full article in the description, and as usual, please like, share, subscribe, and take care.